Good morning, grade 10. We are now in the second module for the third quarter, okay, for weeks 5 to 8, right? And as promised, okay, let's tackle this said module, which entitled DNA and evolution. What's with the DNA and how DNA is really connected to evolution? How this also connected to the human reproductive system, because the first module that we had in this quarter, this third quarter, was the human reproductive system. So that is actually the introductory lesson for the DNA and evolution, right? How do this DNA evolution also continue our generation with regards to the survival of the fittest, okay? The continuation of the strong, the elimination of the weak. It's according to Charles Darwin's theory of evolution. What else? How do this evolution also, okay, affect some organisms, including us, right? Let's continue. DNA and protein synthesis, okay? As we all know, we are actually made up of a lot of proteins. We are also made up of a lot of processes, including protein synthesis, okay? The deoxyribonucleic acid, or DNA, is one of the four macromolecules, okay, essential for life. The DNA is made up of building blocks called nucleotides, okay, which consists of three parts. It has the three parts, okay, a deoxyribose, this is a sugar, okay, a phosphate group, and a nitrogenous base pairs. They are actually group of two, okay, pairs. The nitrogenous bases are grouped into two, okay, two ring purines or purines, which are adenine and guanine, A and G, and the single ring pyrimidines or pyrimidines, which are cytosine and thymine, okay? Pyrimidines or pyrimidines, they're actually the same. The arrangement of these nucleotides, okay, provides the genetic information carried by the chromosomes. Remember that the genetic makeup is actually composed of a lot of nucleotides, okay? DNA contains the sequence that provides all information necessary for proper functioning of an organism, but the actual work of translating the information, because they are actually carrying, your DNA is carrying a lot of information through proteins, and the actual, okay, actual work of translating the information into the material that is used directly by the cell is done by the RNA. This is the reason why, okay, our vaccines for COVID-19, the new technology that we are actually using is RNA-based, mRNA, the messenger RNA, because these are the RNA itself is the one translating, okay, the said information, decoding the information from the said DNA, okay, from these genetic materials, okay, for us to create an antibody against that particular disease. RNA serves the role of being the mediator of DNA and protein, okay, since he is the one, or this is the one, who actually translate or decode the information. Specifically, RNA dictates what proteins are to be synthesized. Correct. Becomes part of ribosomes that creates proteins and moves the amino acids, that's the building block of proteins, to ribosomes. This mechanism involves the production of proteins from DNA, are parts of the processes of protein synthesis. Okay, the synthesis of proteins is the most important event in cells, because not only does it provide protein necessary for structural component of the cell, it also creates enzymes, the enzymatic activity, okay, the enzymes needed to produce the other organic macromolecules. We are actually made up of micro and macromolecules, okay? In 1953, James Watson and Francis Crick unveiled the most accurate description of a three-dimensional double helix structure of the DNA molecule. Remember that if you're going to look at the DNA under the microscope, we cannot actually use an ordinary microscope, just like what I have here. I have an electric microscope, compound electric microscope. We cannot use this one in checking on the DNA, since DNA needs a powerful microscope, like um, the huge microscope that are actually used in the laboratory, okay? Because these are complex, okay? Complex component of a cell. Remember that this three-dimensional double helix structure, okay, has a lot of information that actually make up an organism, okay? Talking about James James Watson and Crick, okay? Again, they actually what? Created a three-dimensional double helix structure, a concrete one, okay? With all the components, the three components of the DNA, and they published their model into two-page paper that eventually became the cornerstone of modern molecular genetics. The features of their DNA model include: one, a double helix or double-stranded helix with two strands connected by hydrogen bonds. Okay, adenine bases are always paired with thymine. A is to T, okay, and cytosine are always paired with guanines, okay, or C is to G, which is consistent in the Chargaff or Chargaff rule that states that the total number of purines in DNA molecules equal to the total number of pyrimidines. The DNA double helix is anti-parallel, meaning to say they are not actually parallel, anti-parallel, which means that five end of the strand is paired with the third end, okay? The fifth end of the strand of one strand is paired with the third end of its complementary strand, and vice versa. So meaning to say, they are not parallel. That what makes them a helix, a double helix, a bond helix, right? This is the anti-parallel strand, which is the fifth and the third, okay, and the third and the fifth, and so on. So vice versa. So the fifth end is actually connected to the third end, making them a what? A helix. Because they're anti-parallel. So they're actually twisted with each other. Okay. Not only are the DNA base pairs connected by hydrogen bonds, take a look at this one. Okay. Thymine is bonded with adenine with two hydrogen bonds, and guanine is bonded with cytosine with three hydrogen bonds. Okay. Let's take a look at later what are the connections of these bonds. Okay. Not only are the DNA base pairs connected by hydrogen bonds, but also the outer edges, the nitrogen-containing bases, are also exposed and are available for hydrogen bonding. Take a look at this one. That this, if you're going to look at this, it actually resembles what? This is usually resembles in three sets. We call them codons. Okay, these codons carries the information necessary for a particular protein to be produced. Okay, they're not the one. Okay? This is not the one. The DNA is not the one who creates. Okay, they're not the one who creates the said proteins. Okay, but they contain the necessary material. They contain the information needed for us, for the body. Okay, for the RNA. Okay, the RNA decodes the information for us to create a particular protein. Okay, RNA. RNA stands for ribonucleic acid. It's named after the 5-carbon sugar group that makes up the backbone, the ribose sugar. Okay, just like DNA, RNA has sugar phosphate backbone with nitrogen base attached to make it up, to make it up a nucleotide. Okay. However, RNA is single and is single-stranded. When you say single-stranded, it doesn't contain any pair. Okay, and does not have thymine. Instead, it has adenine pairs with nitrogen-based uracil. So what is again the pairing? We have adenine to thymine, guanine to cytosine, or thymine to adenine, guanine to cytosine. But in RNA, if the question there in your exam, the question there in your module is asking about the coded, okay, the coded RNA pair. When we say RNA pair, RNA don't have what? Thymine, okay? Instead, adenine is paired with uracil or U, okay? That's what makes RNA unique because it actually contains uracil rather than thymine, okay? RNA may also be found in the nucleus and cytoplasm of the cell. There are 3 kinds of RNA. Take note, okay? This is based on their function. Again, these RNA are named after their purpose, their functions. Okay. The first one is the messenger RNA, the mRNA. Do you know the purpose already? This is to what? To carry the information because this is the messenger. Ribosomal RNA. Okay. This is to transport in the ribosomes. rRNA. Transfer RNA or tRNA. This is for transferring of decoded information. All 3 together with the DNA work to build proteins needed by the body or needed by the organism's body. Okay, these are the 3 types of RNA. Messenger RNA formed like nucleoplasm with DNA as template. Okay, has groups of 3 bases called codons. It has codons same as your DNA. Okay, the function brings code for synthesis of proteins from the DNA in the nucleus to the ribosomes of the cytoplasm. That's what I had discussed a while ago. Okay, transfer RNA or tRNA. This is a clover-shaped structure with 3 exposed bases, same, called anticodons. Okay, from the mRNA. Okay, one end and an amino acid on the other found in the cytoplasm. So what is the function of the transfer RNA? This is to pick up amino acid in the cytoplasm and fits them into the proper place in the growing chain of polypeptide inside the ribosome. Okay, again, to transfer. That's the function of tRNA. How about ribosomal RNA? Again, made in the nucleolus. This is actually inside the nucleus, the dark area of the nucleus, forms important structure component of the ribosome because, again, ribosomal RNA is the one, okay, giving instructions to the ribosome. Again, these RNAs, okay, are named after their functions, as what I am telling you a while ago, right? Are you still with me, Margie and Dimitri? Okay. DNA in its relation to heredity and diversity. When you say heredity, the transfer of genes, and of course, diversity, how diverse or how wide is the spread? How unique are we in the community? So this is actually what in relation to biodiversity and into heredity or the transfer of genes or transfer of hereditary traits. Humans grow and develop according to the instructions in our genes. Okay, a gene is a segment of DNA that may code for protein of your hair, protein of your like insulin, which controls the level of your glucose or sugar in the blood. Okay, the DNA molecule is very long and may contain hundreds or even thousands of genes in a person or in a particular organism. The DNA in your genes is responsible for your uniqueness. That's why we are unique. Even paternal twins or even Siamese twins, okay, they're not the same if you're going to look at them. Okay, it could be their physical appearance or their behavior, which may differ. Okay, have you known some of the twins in real life? Have you seen some of them, twins, or with relatives that are twins? Margie and Dimitri? Okay. Okay, the DNA in your genes is responsible for uniqueness and why you share some features with your family members. Just like, oh, you look like your dad, or you look like your lolo, your lola, and so on. This is because of the transfer of these genes through heredity. However, before the information in your genes becomes useful, the segment of the DNA should be cracked or decoded. Okay? The code dictates the order by which amino acids will be assembled to form a protein. Okay? DNA therefore codes the structure of the protein, and the protein's structure in turn, okay, will determine its role in the body. It must be remembered, though, that all cells must carry the same genetic information, otherwise it will become what? It will become what? If a particular cell didn't actually or different from the rest of the cell, it will become, number one, it will become cyst. Have you heard about cysts? Okay, it will become an abnormal cell, or it will become a cancer cell, because the genetic information in that particular cell is different from the rest of the body. Okay, this is why before a cell divides during mitosis or cell division, the chromosome are doubled so that the daughter cell will have the same copy of the genetic material. Or otherwise, in the case of cancer cells, okay, metastasis will happen. When you see this metastasis, this is the spreading of cancer cells. Replication is a process by which a copy of original genetic information is duplicated so that each new cell receives exactly the same information as of the parent cell. Okay. Again, if that particular cell is different from the rest, okay, it is considered as an abnormal cell. Okay, and there are a lot of causes if in case. The sequence in one strand of DNA will automatically serve as the template to supply the information needed to produce its partner or complementary strand or complementary base, okay, or complementary base pairs. The template and its complementary strand will now make up a new double-stranded DNA identical to its original strand, okay? For the replication to occur, the twisted DNA must unwind and separate. Because remember that this is intertwined. Okay? It should be unwind, it should be removed, it should be separated. Okay? This is made possible by breaking the hydrogen bond. Remember the bonding that I am telling you a while ago? Okay? That adenine and thymine has 2 hydrogen bonds, while cytosine and guanine has 3 hydrogen bonds. They need to be break. Okay? How can this hydrogen bond break? Okay? Between the paired nitrogen bonds with the aid of enzymes. In order for these bonds to be broken, it needs an enzyme to break those bonds, those hydrogen bonds. Free nucleotides in the cell find the complementary base pairs, and enzymes will again bind these nucleotides to make a complementary strand. Okay, copying the recipe. We are now in the case of after protein synthesis, we are now carrying or copying the complete set of genes. Okay, the recipe for cooking protein needed by the body is only confined in the nucleus of the cell. The nucleus also serves as the protective barrier to DNA. Okay, this DNA therefore must be copied because there are many molecules that can damage the DNA in the cytoplasm. There are, of course. Okay, and the machinery to make protein found in the cytoplasm as well. What is the machinery in the cytoplasm that creates protein? Are you still with me, Margie and Dimitri? What is the protein factory of the cell? What is the protein factory of the cell? Margie and Dimitri? We are done with this, right? Parts of the cell, the organelles. What organelle is considered as the protein factory of the cell? What organelle? Can anybody tell me what organelle is considered as the protein factory of the cell? It is the ribosome. Yes, correct. It's the ribosome. Okay, that's the protein factory of the cell. Okay, there are two types of ribosomes. Okay, again, these ribosomes, they're named after they are actually located. Okay, the free ribosomes are in the cytoplasm, and the attached ribosomes are in the ERs. Okay, that's why it is considered as rough endoplasmic reticulum. What makes it rough? Because there are a lot of attached ribosomes. Proteins are made up of chains of amino acid called polypeptides. Okay, the transfer of information from DNA to protein is called gene expression. Here you can actually see the expression of a particular gene if there is a complete transfer of information. It occurs in two steps. Okay, first step is the transcription phase. The second is the translation phase. Okay, let's take a look at the transcription. Transcription rewrites the genetic code in the DNA into the messenger RNA. Remember the messenger RNA? Again, they're named after the function. It works just like DNA replication and follows the base pairing principle, except that adenine in DNA is actually, if you're talking about RNA, okay, except for adenine is being paired with uracil instead of thymine. Okay, when transcription is finished, the DNA recoils to its double helix structure. Again, in the RNA sequence, instead of pairing adenine with thymine, we pair adenine with uracil or U. Okay, the mRNA is now able to leave the nucleus by way of pores in the nuclear membrane. Okay, the mRNA carries the code to the waiting ribosome in the cytoplasm. Okay, remember that your ribosomes are the one responsible in creating what? In creating the proteins needed by the body or the organism. Ribosomes are the site of protein synthesis. Okay, the mRNA possesses the code that specifies the order in which amino acids will be joined to form a polypeptide. The second phase is the translation. Okay, this is the second step in gene expression. Okay, whereby codons, these are the consecutive 3 nitrogen base pairs, okay, or bases, are read and translated into amino acids. This is the time now that we actually decode. If mRNA carries the information in the transcription phase, in the translation phase, this is now we translated the code, okay, in a particular protein. Ribosomes consist of ribosomal RNA or rRNA, again, based on the function. A functional ribosome consists of 2 subunits. we have the large and the small subunits. The mRNA enters a ribosome, okay? Since the code in the mRNA does not have the punctuation mark that might signal the start and end of the codon, okay? There must be a particular starting point, the start here codon. So what is the start here codon? To begin with the translation process so that the whole sequence will be read correctly. Okay, this codon is initiation codon. Okay, what is the initiation codon? We have AUG. Okay, adenine, uracil, and guanine. And in mRNA, that also codes the amino acid to methionine or AUG. Okay, adenine, uracil, guanine. I will let you see the table later of these codons. This set of codons, or this set of three bases has a particular or has its own what? Has its own protein. Okay, methionine is one of them. Methionine is the initiation codon. This is the start codon. We usually have the start and we usually have the end. What if there is no end? We started with AUG and there is no end codon. What will happen to the cell? Will anybody tell me what will happen to the cell if there is no stop codon? We started with the initiation codon, which is the methionine. But what if there is no stop codon? What will happen? What will happen? Of course, the ribosome will continue to create a lot of proteins that are not necessary, that are not needed in the body. And this is what makes, again, the cell abnormal. We are creating abnormal cell with these ribosomes. Okay, we're creating abnormal growth of cell because of this ribosomes. Okay, let's continue. The tRNA picks up the amino acid methionine according to their triplet base or bases, or the anticodon UAC. Why UAC? Okay, let's take a look at this one. We started with AUG, adenine, uracil, and guanine. Okay, so the anticodon for this AUG is adenine is paired with thymine. Okay, but in the case of RNA, instead of thymine, we have uracil. Uracil is paired with adenine. Okay, and guanine is paired with cytosine. So this is the anticodon of AUG. Okay, the UAC. This is the opposite of the mRNA codon attached to one end of tRNA. Okay, the anticodon of tRNA, UAC, binds with AUG at the P, or the site of the large subunit of the ribosome. The next mRNA codon is lined up at the A site. Okay, the sequence repeats the polypeptide chain until the ribosome reads any of the stop codons. We have stop codons. Okay, these are the stop codons. Please take note of the stop codon. We have UAA, uracil adenine adenine, uracil guanine adenine, or uracil adenine guanine. Okay, please take note of this because this will come out in your exam. Okay, this will come out also in your activities. A lot of your activities are with this. If in case you miss this class, this online class, I will upload this immediately in the LMS so that you can review. Okay, I'm stressing this out, right? That the starting codon, which is methionine, is AUG. And of course, the anticodon for AUG is UAC. Okay, and so on. That's the P site. The next would be the A site. Okay, the A site, this is the site where what? Where all the genetic information, okay, all the lineup pairs are actually in until it reaches what? The stop codons. What are these stop codons? This is to illustrate that the transcription or the translation needs to be stopped. Okay? Needs to stop. Otherwise, we will be creating an abnormal cell. Okay? What are these stop codons? We have UAA, okay? UGA, okay? UAG. I repeat. Uracil adenine adenine, uracil guanine adenine, and uracil adenine guanine. Okay? Am I making this clear? We have less than 1 minute. Let's continue by next meeting. Am I making this clear? Do you have any questions so far? Question. Again, as early as now, please take note of the start codon or starting codon and the end codon or the stop codons. Okay? Initiation codon and the stop codons. Question from Margie and Demetri? Okay. I think that will go for today, class, because we have less than a minute. Thank you for your participation, Margie and Demetri. Please inform your classmates that I'll be uploading this one immediately in the LMS so that they can review, because this is, for me, it's a very important topic. Okay, this is a base topic. Why? You'll be dealing